Hello, I'm Pamela Douglas, and I'm here to talk about computational models to simulate and emulate cognitive processes. So now it's not a surprise, but happy birthday to EEG. This was already brought up by our first keynote speaker. Yes, I can actually do the math, but it turns out that the first demonstration of human EEG was actually 95 years ago, and it wasn't actually published until 5 years after that. So thank you to Hans Berger. Yay. So since that time, we've actually mapped all different types of EEG signatures to different tasks. The readiness potential, music, language, even insomnia was mapped in the 70s, which you might have if you traveled here from, say, California and have jet lag. And similarly, for at least the past 25 years, human brain mapping and connectivity models have linked all different types of tasks to different parts of the brain. But more recently, we have these amazing new open science big data initiatives. So one of them is spearheaded by Paul Thompson and many, many, many collaborators. Some of you are probably part of this, which is the Enigma data set. I was very excited to take part in the Enigma ADHD working group. And one of the interesting things that we learned through this large, mega cross-sectional analysis was that we confirmed this ADHD theory of delayed maturation in the brain. And then more recently, on a smaller yet still very large data set, on the UCLA SIDAR ADHD data set, we went ahead and replicated this same finding, which is the delay of maturation, but we extended it a bit because we showed that it seems to be an asymmetric delay in development across the hemispheres. But what was actually really striking was not just the delay in maturation, but also the delay in degradation. So I found this to be quite striking. In a number of parts of the brain, there was actually a delay in neurodegeneration later in life in the ADHD group. So if you take that together with the fact that psychostimulant drugs actually help normalize the changes in the ADHD brain, it perhaps calls into question the extended use of psychostimulant drugs for ADHD children. So these big data sets have shown enormous potential. We've learned a ton using them with higher statistical power. But unfortunately, brain maps alone do not reveal computational mechanisms in the brain. But of course, as Jennifer just said, brain maps do constrain theory because, as we would expect, information exchange incurs energetic costs that scale with distance between regions. And so therefore, we would expect regions that need to communicate with higher bandwidth and shorter latency to be located closer together in the brain. So once again, the same theme that topology constrains function. But this problem of brain mapping not really answering questions was essentially pointed out in the 70s by Alan Newell, who was frustrated with the field of cognitive psychology because experimentalists were in the habit of testing one hypothesis at a time. And this, of course, leads to this reductionism and eventually to just paradigm-bound theories. So it doesn't really synthesize the information in a useful way if you're trying to learn about the algorithms of the brain. So if the goal is to then integrate this knowledge into a unified model of brain information processing, we should try to create a model that can accomplish cognitive tasks using neurobiologically plausible subcomponents, also model or generate dynamic neural activity data, and also this last problem of determining how different sub-functions in the brain are deployed. So this brings together three different fields: computational neuroscience, cognitive science, and artificial intelligence. So one way to conceptualize this is to think about this in terms of Mars' three levels of analysis: the hardware, the representation, and the computational level. So from this point of view, cognitive science has taken a top-down approach where they've been unencumbered by the need to try and explain brain activity data and try to use neurobiologically plausible subcomponents. They've just tried to explain human behavior data and try to explain behavioral data on tasks. Conversely, computational neuroscientists have taken this bottom-up approach where they've tried to build biologically plausible models that then generate and recapitulate dynamic neural activity data. So one of the things that helped inspire these neurobiologically plausible computational neuroscience models was this early observation that there were these stereotyped patterns in different cortical columns all throughout the brain. So these repeated motifs seem to just be repeated all throughout the cortex. So the early models, the mean field and neural mass models, such as the one developed by Wilson and Cowan in 1972, they were limited by computational power at the time, so they just had a single inhibitory and a single excitatory cell, which represented population activity of those two subtypes of neurons. But as computational power grew, Douglas and Martin, not this Douglas, a different Douglas, added laminar specificity, and later, of course, the canonical microcircuit model also added local reoccurrence. But despite the simplicity, this Wilson and Cowan model was also very useful because it predicted this need for a tight excitation-inhibition balance in the neural code. And we know this to be extremely important because when it's disrupted, we actually get seizure activity. So even though these computational neuroscience models are very useful, they generally are not used to perform cognitive tasks or to explain behavioral data. So I started to think about this problem. If the hardware is actually the same, how do we have such diverse functions in the brain? So I found this paper, which I think is quite interesting, from NeurIPS this year, where they started with two different initializations of the VGG16 deep neural net. So the exact same structure, but they seeded it with different initializations on the weights, and they also trained it with different data. And what you ended up getting was a very similar accuracy across both networks, but very different representations. And it turns out that the dissimilarity of the intermediate patterns increased along the processing layer. So I found this to be quite interesting, that you can actually have the exact same architecture, but it learns different representations. So of course, understanding representations is quite important. So this is this nice paper by Alex Binder and colleagues, where they were trying to classify airplanes, bicycles, birds, boats, and they had a very nice accuracy on horses using either Fisher vectors or deep neural nets. But they wanted to understand the representations that were driving that classification. And it turned out that upon closer inspection, using this credit assignment technique, known as layer-wise relevance propagation, there was actually a copyright symbol in the lower corner of all of the horse pictures. So they thought that they were doing really well on the classification, but the Fisher vector actually just learned to classify based on the copyright tag. So that's just one case where perhaps you should be careful about what you're classifying because you want to understand what features are driving the classification. So coming to this level of representation, of course in neuroimaging, there's two classic techniques that we use most often to study representations in the brain. Those would be encoding and decoding models. So they work in a nice complementary fashion where encoding models try to take aspects of the stimulus and predict the representations in the brain. And conversely, decoding models try to take the representations in the brain data and make predictions about the stimulus, or occasionally about behavioral responses. So these two can actually work together in a complementary way along a hierarchical pattern as well. And there's the possibility of having many, many different causal pathways of encoding and decoding along a hierarchical message passing pathway. So I found this and I thought this was quite funny on a blog. Why would you try and predict the task? We already know what they're doing. This is for an unknown reviewer many years ago. But of course, what we're trying to do is learn about the representations. How is the brain representing the stimulus? How is it learning those representations? So that's part of course, the goal of understanding decoding models. is to reveal the representational content and the fine-grained patterns, and to try and understand if the neural code does exist in perhaps Apache functional segregation. One of the most impressive types of decoding models is this ability to sometimes reconstruct stimuli, which is a quite impressive feat of decoding models. So of course, there are many, many, many different types of classifiers, each with their own inductive bias. But linear classifiers have just dominated in terms of their use, mostly in the fMRI and EEG worlds. But why? Of course, they're computationally efficient, but there's this idea that they're also very easily interpreted. And also, linear classifiers are thought to work best at the level of readout, therefore detecting explicit representations. So the notion of perhaps cars versus vans is thought to be hopelessly entangled at the level of V1, but as it processes through the visual stream, it becomes untangled and then can be classified using linear components. For me, it would always stay entangled because I have the equivalent of prosopagnosia, but with cars. So, so of course this is true. This is some nice work by Hong et al. That showed that the performance of linear decoders does increase for processing object category. But unfortunately, even linear classifiers are very difficult to interpret. This was pointed out by Stefan Haufe and many years ago by Isabella Guyon. The weight vector that we get from a classifier is orthogonal to the decision boundary. And if we have covariance between the noise voxel and the informative voxel, we can get this situation where it turns out that the weight assigned to the noise voxel is actually stronger than the weight assigned to the informative voxel. So there's this nice corrective technique that Stefan Haufe pointed out, and it works quite well, especially if the noise is Gaussian and everything works out very nicely. But unfortunately, there's different situations when the noise is non-Gaussian or if there's a lot of redundant voxels. And in that case, the recovery technique doesn't actually work. And unfortunately, regularization does not help the interpretability issue because L1 penalties will somewhat randomly assign weights to the informative voxels. So correlated informative features, some of them will randomly appear to have a very small weight. So if you put all of this together, presenting weight maps may in some sense lead to an implied interpretation. In fact, you could actually present something which looks to be like Apache functional organization when in fact the underlying signal is much more structured. But this is only in the case when your features contain mutual information. But what if the features are independent? So I've done a lot of work with Ariana Anderson and Mark Cohen using independent components as classifier dimensions. And we've found, which is consistent with a lot of interesting work that I've seen at this meeting, that you can actually achieve very high accuracy using only a few dimensions. So because of this, independent component maps or principal components, which I saw yesterday, can be interpreted with a lot more ease than this just using voxels as features. So it turns out that encoding models, which take aspects of the stimulus and try to predict the response, also have a similar problem. So you could perhaps take aspects of a scene or sounds and try and predict tonotopic maps or activation in the visual stream. And different feature sets may be useful for making different predictions along the visual pathway. But it turns out that it's possible to have multiple different types of features in an encoding model that all explain the data equally well. So for some reason, it didn't go to the, there we go. Here's an example from Diedrichsen that showed that principal components or finger synergies or even just random vectors could all explain the activity in the motor cortex equally well. So in this sense, you may, if you just test one model, get this single model fallacy. where it would appear as though you've explained the data quite well, and one might infer that those features are actually important for driving the representations that you measure. So if you're a systems ID type of person, then this is analogous to a problem which is non-uniquely identifiable. So it's always important to test multiple models and use the data to adjudicate between competing models. So I don't have time because I'm over time already, but moving forward, there's a lot of new work using neural network models, reinforcement learning, and Bayesian models to study representations in the brain. And there's many different parallels, and many of these internal representations are useful for modeling and making comparisons to the representations in the visual stream, for example. So just to summarize, traditionally there's been this trade-off between biological and cognitive fidelity, but we can turn those into a synergy if biologically inspired models can achieve cognitive feats. But of course, this will take collaboration between lots of different labs and an open science culture. So bringing all of these together, we can eventually build brain computational models, and hopefully we can then harness the power of all of these big data sets to test out different brain computational models and perhaps use transfer learning to generalize across time between subjects and across tasks. So thank you so much for everyone for having me be here. Thank you to Nico Kriegeskorte, and our amazing next speaker is Alex Gramfort.